Okay, so genetics of blood introduces two new things, multiple alleles and codominance. So we were introduced to alleles last week, and we know they're different forms of a gene. So multiple alleles maybe gives you the idea that there's many different alleles for a single trait. And then codominance, we learned dominance will show no matter what, or I usually use the word expressed. So it'll be expressed no matter what. So to say that it's codominant, maybe you're thinking coexisting, cooperating. So co means go together. So in that case, there'll be more than one allele that will be dominant. So our first example is that of the roan horse. So the roan horse is probably like a classical example for codominance. So it would have had a white horse, like a white-haired mother and a chestnut-haired mother or father, maybe. Sorry. And when they come together, they mix their alleles. So it inherited one of each. Now, if white was dominant, the horse would only be white. If chestnut was dominant, actually that would be bay. The reddish color is bay. Then the bay would be dominant. So then she's not red, she's not white, she's both. And so this is codominance where both alleles will be expressed. Some of the hairs are white, some of the hairs are red. Different than like incomplete, which we're not doing. Incomplete dominance is where they would blend together. So this horse would have been pink, but he's not. Okay, so with related to blood, a common example of codominance is sickle cell disease. So here's some sickle cells, and you can see you have normal cells and you have sickle cells. So that's showing you both alleles are being expressed at the same time. Some got the allele for the regular shape and some got the allele for an awkward shape. So sickle cell disease, and we use AS, we usually use different letters when we're doing codominance to show one is not dominant, one is not recessive. So A is for the normal allele and S is for the sickle cell allele. And a person who has AS has a little bit of each. Oh, blood cells didn't finish there. So it has some normal cells and some sickle cells, and that's because the protein that it's coding for is hemoglobin, and the normal hemoglobin should not clump together. But a sickle cell anemia happens because, or sickle cells happen because these hemoglobin molecules stick together. Now, hemoglobin is supposed to carry oxygen, and oxygen, you know, we use for all of our energy, basically. So people who have sickle cells, they're not able to carry as much oxygen. And also, if you look at this vessel, single cells fit through just right, but a sickle cell will clump together and it gets clogged in your smaller vessels. So if you have like homozygous sickle cells, and that means all you have are the sickling cells, then you have a lot of pain, low energy, things like that. If you have heterozygous, you still have some functioning cells, and so you can lead a relatively normal life. So it's really a concern when you get the homozygous S allele. And that can be painful, cause poor circulation, jaundice. Anemia is just a lack of oxygen, which can happen for a number of reasons, and also hemorrhaging, and that means bleeding. Heterozygote carriers usually don't show these characteristics, but they do have the sickle cells along with the regular cells, so it's said that they have sickle cell trait. So homozygous for sickle cell is sickle cell disease, and heterozygous is sickle cell trait. So these two individuals here have sickle cell traits, and they have 4 babies. And if we did a Punnett Square, we would find like that 1 to 2 to 1 ratio that we were seeing last week with the purple flowers. So 1 was homozygous, what we would have said is dominant. 2 were heterozygous, and 1 is homozygous recessive, if you will. But in truth, 1 is not dominant, 1 is not recessive, because they're both being expressed in the heterozygous form. Now, it's more common in Africa and areas where there is malaria because this heterozygous individual is actually protected from malaria. And so with evolution and survival of the sickest, or the survival of the fittest, sorry, then that trait survives, is passed on to future generations. So you can see like 3 of these individuals are going to get the sickle cell allele. So 3 out of 4 means that that allele will be more common. So people had a couple people had trouble just working with these diagrams or this picture, so I thought I would help you. So the woman has sickle cell trait. So does that mean she's homozygous or heterozygous? Sickle cell trait. Remember, disease is homozygous and trait is heterozygous. So it's saying that she's heterozygous and her husband is not. So that's, I maybe needed more words in that. It really means he has totally normal genes. So to complete the Punnett Square, I double click my drawing, and then I need to select the letters. So I'm selecting letters. Some of you have figured this out, some of us haven't. So I can move my letters around. Woo! Okay. So if he is normal, he has A and A. And then if she has sickle cell, she has A and S. She has sickle cell trait. So her letters will come across, his letters will come down. So this individual, whoops, well, I'll work with it later. This first individual will be an AA. This second individual will be AA. And then let's push them up. Then if this one comes down and this one comes across, this person is AS. And this person is AS as well. So that's how you can edit that. That would be a capital S. We don't use lowercase when we do codominance. Okay. So now that I've edited that, the question is, what is the likelihood that their third child will be completely normal? So homozygous A. So what do you think from this square, which somehow shrunk when I did it? 2 of the boxes were homozygous A, 2 of the boxes were heterozygous. So in a fraction form, what would be my answer? 1/2, 2 out of 4. So it doesn't matter if every child has the trait. Each child has the Punnett Square possibility, right? And it's, as we learned with our flipping of the coin, it's not guaranteed that it's going to come out in that fraction. But if we looked at a very large population, as we did with the whole group data, we'll get much closer to that trend. Okay? All right, so back to the PowerPoint. In the PowerPoint, I'm covering all like the informational piece that you get in that first page and informational pieces that you get along the way. So you can probably jump to just answering questions after setting through the PowerPoint. So that was codominant, simply that the 2 are expressed at the same time. So now we're going to talk about multiple alleles. So far we've only talked about 2 alleles, like big P, little p, A and S. Now we're going to talk about when there's more than 2 alleles that code for a trait. So, and the classical example is the rabbit's fur color. So they have 3 or 4 different alleles, and depending on the combinations, you get different colors. And so that's a good example of multiple alleles. I should have put their letters up. Anytime you have something that's non-Mendelian, like multiple alleles, incomplete dominance, codominance, you'll get more than 2 variations in the phenotypic expression. So like here, there's 4 different examples. Blood type is another one. So blood type is governed by 3 alleles, and they're the A, B, and O, but we don't type, like the allele itself is not written formally as A. It's I subscript A, or superscript, and I superscript B. The I stands for immunoglobulin, which you heard in that video. causes an immune response. So you can have the A allele and produce proteins that are of a specific shape, and they're on the surface of the blood cell, or B, and it produces a different shaped protein, or I, which is O, the lowercase I would tell you it's recessive. So in this case, A or B can mask O. Okay, so allele A, B, and O is written like classically IA, IB, and little I. So looking at these, you can see the shapes on the red blood cells. They're different shapes, A and B. AB is an example of codominance like your sickle cell. Both are expressed, so this cell will have both shapes, the A shape and the B shape. O doesn't have either of those two shapes. It does have other marker proteins on its surface, but they're not ones that cause immune response, so we don't concern ourselves with them in genetics. Okay, so they are codominant with each other, but they are dominant over I. And you can refer to these as marker proteins, proteins, glycoproteins. You'll hear them named different things, but basically they're proteins that are on the surface of the cell. So here's the genotypes that you can have for the different phenotypes. So how it's expressed physically is the blood type. The genotype is the type of genes you have. So IA would be homozygous for type A. IA little I would be heterozygous for type A. So anytime they're heterozygous, they have one of the recessive alleles. AB is codominant. There's only one way you can get that. And O, there's only one way you can get that. You'd have to be homozygous recessive. So I should add another IA to this right here. The genotype would be IA or IA little I. And you have on your worksheet, I believe, if I scroll down, you have a chart to fill out such a thing here. So like IA, if you scroll down to number 2 on page 4, IA would have what type of blood? And little I would have what type of blood? I should actually, so you would have wrote A and O. Okay. You could go here. IB codes for which alleles or which proteins. Will they have A? No. Will they have B? Yes. IBI, will they have A? Will they have B? They will not have A, they will have B. Will AB have A? Will they have B? So if they have the IA allele, they'll get the IA protein. If they have the IB allele, they'll get the IB protein. Okay. So back to the PPT. Whoa. Whoa, sugar jets. Hold your horses. Okay, so this is showing you that same thing, the different proteins that are on the surface. If it's A, it's got this circle. If it's B, it's got this diamond. If it's AB, it has both. And if it's O, it has neither. Now, I'm not going to get too far into the antibodies, but your body has the antibodies for the antigens you lack. So if you have the A antigen, you don't want an A antibody or you'll be attacking your own blood, right? So your body recognizes the A antigen as self. And if it were introduced to a B-type blood, it would recognize this diamond as not belonging. It would treat it the same way as it treats a disease. So the anti-B would attack the B antigen, anti against B. So if you have A blood, you have the B antibody. If you have B blood, you have the A antibody. If you have AB, you have neither, or you would attack your blood. If you have O, and Ray, this is why O can only receive O, because they have both antibodies. Okay, so let's try one together. So you might have started working on your worksheet, but if I could have you with me in the chat room, otherwise I sit here and wonder where you are. So we're going to do a Punnett Square just to make sure you're good to go with the Punnett squaring. Dad is heterozygous for type A, so what genotype does that mean he has? Mom has type O blood, so what genotype does that mean she has? What are the possible blood types of their children? On your mark, get set, go. I'm going to pull up my chat box and I'm looking for some answers in my chat box for the next couple of slides. So if you do the Punnett Square, what blood types does the children have? So first you'd have to figure out dad's genotype. If he's heterozygous, it means he has a dominant and a recessive allele. If you figure out mom's genotype, she's type O, which is recessive. The only way you can show a recessive gene or trait is to be homozygous recessive. So type in the chat box, what type of blood will their children have? Bring dad's alleles down, mom's alleles across. Some will have type A and the others. Good, okay, and 50% for the others. Okay, so we understand how to do this. We're gonna do one more and that's gonna be it. Here, this one is a little bit of a different approach. We don't have all the information, so there's more to solve. A man has type A blood, marries a woman with type B blood. So remember, there's two different genotypes. Thank you, Ray, half of each. There's two different genotypes that code for A or B. Their child has type O blood. So this one is kind of a backwards working. You know the offspring. So now you have to work from the inside to the outside of the box to figure out what are the parents' genotypes. And I don't have a slow reveal on this one. So see if you can figure it out. So for mom to have type A, or man, the father to have type A, would he have AA or would he have AO? In order for the woman to have type B blood, she could be homozygous, IB, or she could be heterozygous, IBI. Their children have type O blood, and there's only one combination that would allow that to happen. So what are the genotypes of the parents? And what's the phenotypes of the other children possible? So if you put at least an A and a B outside of the box, but Lauren, what's the genotype? So you need two alleles for a genotype. So are they IA, AA in other words, or are they IA little I, in other words, IO? And the same thing with the type B. Would they be IB, in other words, BB, or IB little I, in other words, BO? BO is always funny, right? Because it's like body odor, BO. Oh, my order is out, it's out of order. Yeah, good. In order for the O to show up, you would have to be heterozygous for each parent. So normally I would have just put in the IA question mark, IB question mark, because that's the information I knew. And then homozygous for O, I knew was one of the children. So the rest of this I wouldn't have filled in yet. So I'd fill in 1, 2, 3. Those were the known pieces of information. Now I can work out of the box. I know this I had to have come from one of the parents, so I place it above. This I had to come from the other parent, so I place it out to the side. So now I know it's heterozygous. So the phenotypes, blood types, so you have one that's going to be AB, one that's going to be A, this one should be B, I got to fix that typo, and one that has O. So you have one of each. Okay, so in the chat type box, or this might be our last slide, gang. So in the chat box, I got some questions for you. What genotypes code for type A blood? There are two genotypes possible. What are they? Type it in. What genotypes code for type B blood? Remember, they need two alleles. Good job, Ray. Good, Kiara. You got the homozygous and the heterozygous. Sophie, is this Sophie M, I think, or is it Sophie G? Yes, very good, Sophie. And AB and O. You guys got it? Yep, you got it. So you got the hang of this. So you have to recognize the different genotypes if you're going to do a Punnett square, because remember, the genotypes of the parents are what you're putting on the outside. So this little chart helps you. These are the phenotypes in color. So type A blood, type B blood, type AB. And then the 2-letter combination above, that's your genotype. So I think we had a similar question on our worksheet. Here it is. On page 5, number 7, asks for genotypes and then gametes. So you guys already told me, I'm going to go back to your answer for genotypes possible. So you had IA or IA little I. And I won't bother superscripting right now, but remember, gametes is when they split in half. And really what you're putting up here is the parents' gametes. So to say IA, you would put one IA here and one IA here. Or to say IA little I, just what you have here, right? These are the parents' gametes. These are the children's zygotes. So the possible gametes, depending on their phenotype, they could make an IA gamete, or they could make an I, lowercase I gamete. Because remember, meiosis and segregation, that IAI, they're going to separate, those homologous chromosomes will separate during formation. In the end, you're using everything you learned to decide if these children have the right parents. So multiple alleles, blood typing, you have more than 2 genes. Codominance, both of these express codominance where there are 2 versions being expressed at the same time. So do you guys have any questions for me? And hopefully we already got you started on that worksheet, so part of the work is already done. Till next week, and we start talking about how genes actually are expressed as proteins.